Hi, everyone. We're going to wait a few minutes to give everybody a chance to join, and then we'll get started shortly. All right, if everybody's ready, we're going to go ahead and get started. Hello, everyone. Thank you for participating in today's Zoom meeting on trial expectations, what to expect. Today's guest speaker is Jacose Bell. Jacose is the Senior Patient Advocacy Lead at Spark Therapeutics. Jacose joined Spark in April of 2021 and has had the privilege of supporting relationships and partnerships with the greater Pompe community ever since. She is passionate about raising awareness and providing education about AAV gene therapy as part of that role. During the presentation, you can send questions at any time by clicking the Q&A feature at the bottom of the Zoom window. The guest speaker will answer the questions at the end of the presentation. Thank you very much. And with that, I'll turn it over to Jacose.
Thanks so much, Tiffany. Hello, everyone. Good morning, afternoon, or evening, depending upon where you're joining from. I'm going to go ahead and share my screen and get started. I want to jump right in since I want to make sure to leave plenty of time for a conversation. So just want to confirm if somebody could, you can see my screen. Great. Okay, so let me just make sure. I'm going to go ahead and go off video just so as to not distract while I'm presenting. So we're going to talk about adeno-associated virus gene therapy research and how investigational AAV gene research trials are conducted and things you want to think about and consider. So I want to share a little bit about Spark Therapeutics quickly. So Spark was founded, we're based here in Philadelphia, which is where I join you from today, in March of 2013. And we're really focused specifically and only on AAV gene therapy and genetic disease. And we are, as part of that, very committed to education. So again, thank you for having me. We have programs, as you can see along the bottom, in inherited retinal diseases, lysosomal storage disorders, of course, like Pompe, neurodegenerative disorders, and certainly are always looking for other areas of genetic disease that we may be able to support. So today, we are going to understand a bit more quickly about AAV gene therapy research. I'll move through some of that fairly swiftly, but as this will also be archived, you'll be able to go back through and watch and review the slides. And of course, if you have questions about AAV as a technology itself, please bring those to the Q&A. And then we're going to talk about investigational gene therapy and its different approaches. And finally, spend a good amount of time on becoming more familiar with investigational gene therapy clinical trials and how we think about clinical research in this field. Oh, I did that already. There we go. But first, just a few questions that you don't need to necessarily answer, but maybe just to sort of warm us up, you can consider, and you're welcome to drop it in the chat if you'd like. And the first is that I want to understand a bit about whether or not you have heard of AAV gene therapy research before. I'm going to guess, just by having taken a quick peek at our participant list for today, that many of you have, right? And certainly where you may have heard about that is also going to be of interest. But getting a little bit of a sense of, have you heard about gene therapy research before? And if you're willing to share in the chat, where you think you fall on this spectrum. Yes, I've heard of it, but I don't really know a lot about it, all the way through to I have extensive knowledge and feel really comfortable. And I can see that we're getting some folks chiming in. Thanks, Allison. Thanks, Anik. Yes. So some people, some familiarity, a little bit, but good. Well, we're going to talk about it. So hopefully by the end of our time today, you're going to feel like you know a little bit more. And sorry, just trying to get my slides. There we go. And so for those of you that have heard about gene therapy research I would love to know a little bit more about where. And you can choose any that apply here, and also just say you can drop that in the chat. So you heard about it through a doctor or someone on your care team. You heard about it at a conference, an event, something research focused, newsletters, websites, all of the above. B, C, and D, great. Yeah, so I'm glad to know that you are hearing about opportunities, and I'm sure you are hearing about them at conferences, through newsletters, through opportunities like this and others that AMDA and your patient advocacy groups provide, and certainly through your own research. That's great. So I'm going to skip through this just in the interest of time. Gene therapy research tends to bring up a lot of feelings for folks, so maybe we'll save some of this for conversation as part of the Q&A. But be ready to maybe talk about how you're feeling about the idea of gene therapy research as we get to the end of our the end of this and get to jump into a conversation. So I'm going to start with a pretty lightning quick round on review of genetics, just to sort of reground us in where this science comes from and what we're trying to address. So genes are, we like to say, the body's instruction manual. And so we really like to, at Spark, use metaphors to kind of help make these concepts a little bit more approachable. But we tend to start by first saying, right, your body's made up of trillions of cells, literally trillions with a T. And at the center of each of those is a nucleus within each of your cells. And your nucleus is kind of like a reference library. It's going to have all of the instructions that your body needs to function. And those instructions are stored on chromosomes. You have a total of 23 pairs of chromosomes. That's 46 in all. And your chromosomes are like the individual books or how-to encyclopedias inside that reference library. So each chromosome is like a reference book inside that reference library. And within your chromosomes, you have DNA, right? Deoxyribonucleic acid is what DNA stands for. And that is really the alphabet that your body uses to kind of write those instructions with. DNA is then organized into genes, and genes are little small sets of instructions. I tend to think of them sometimes as recipe cards or specific chapters within some of those reference books that I just mentioned, right? But they're the very specific sets of instructions that your body uses to make all sorts of enzymes and proteins, which then go on to build and regulate and maintain every single process in your body. Oops, sorry, everybody. So there's just a visual representation of that metaphor that I took you through, right? The nucleus being the reference library, your chromosomes, each representing kind of an encyclopedia of sorts within that reference library. The DNA being the alphabet by which those instructions are written into the books. And those instructions, those various chapters and sections being the way that your body then knows how to make all of the proteins that regulate all the functions of your body. So when we are looking at trying to solve genetic diseases, first, let's just quickly run through what's happening, right? In what we would call normal functioning within a gene, we have those little sets of instructions that are used to sort of build all of these various proteins. And most of the time, right, they come out exactly written as they're supposed to. But sometimes there are changes that occur, and a lot of times those changes are benign. There's all sorts of ways that some of our functional genes may be written or changed slightly that don't cause any sort of disease or issue that we are discernibly aware of. But sometimes, as you all well know in this community, one little mutation or a family of little mutations, right, can lead to dysfunction in the way those proteins are created. So you have either limited production, you have no production, and you end up with reduced or really deficient protein activity in the body. And obviously that's what's going on in Pompe disease. So what are we hoping to do to address all of this with AAV gene therapy? At the simplest level, investigational AAV gene therapy can be described as really a therapy that's hoping to deliver genetic material using adeno-associated virus vectors to treat genetic disease. And that is not a simple sentence. So I'm going to break that down over the next few slides, and we're going to talk about what that means. But we have a lot of hope with AAV that it has the potential to address genetic disease at its source, at that gene level of where the problem is, and that it has also the potential then to treat a lot of different types of disease, whether that's lysosomal storage disorders, but also things like cardiovascular disease, neurodegenerative disease, vision disorders, musculoskeletal diseases. So the potential of these therapies is pretty tremendous. So very quickly, I'm going to take you all through the three kind of general categories of how gene therapy is currently being researched. So it's a field that is evolving daily, right? But that generally involves these three modalities. You have AAV gene therapy or gene transfer therapy generally, which is this sort of first panel where you'll see that arm with the infused material going in. Then we have gene editing, and then we have cell therapy. So in gene therapy, essentially the process involves introducing a new functional copy of a gene with the aim of that new copy being able to restore or enhance normal function of a protein within the body. So we use AAV as a vector, which is essentially a delivery vehicle. A vector is a way for us to deliver that new gene to certain types of cells in the body. And we do that by infusing that, as you can see, where it's going to head to your cells and to your cell nucleus, delivering that new little set of instructions to your cell so that your cell then knows how to make those proteins. You've likely also heard of gene editing, and I think the most common example is CRISPR or Cas9 that you've probably heard of in the news. And in this technique, you're not adding in a piece of genetic material that's been created outside of the body. You are actually adding, removing, or changing existing DNA within a person. So those little scissors, right, that you see are often kind of a visual association with CRISPR or Cas9, because CRISPR and Cas9 literally does edit your own genetic material. It's going to go in and make changes. And some of what we're trying to understand in the research of gene editing is how that technique may affect or lead to other downstream mutations, where we might need to be watching for unintended effects that we can't anticipate. But there's a lot of promising potential in gene editing, and it's being looked at in a lot of indications right now in clinical research. We also have cell therapy, and cell therapy involves transferring cells that are capable of performing a desired or needed function into your body. And cells are isolated first from either a donor, so that's usually somebody that's either closely related to you or somebody through like a donor registry that is identified to be a match with you, or it could be your own cells from your body that are harvested, collected in a lab, and they're modified in the lab, sometimes using gene therapy or gene editing as the technique in the lab. And then those new cells who have been in some way engineered to express typically a new gene are then grown in sufficient numbers within the lab to then be given back to a participant. And probably one of the most common ways that you've heard of cell therapy at this point in the media would be CAR T cell therapy, which is currently being used to treat cancers, certain types of cancers. There are also considerations with cell therapy in that I think, right now, based on where the technology is, there's a somewhat limited repertoire of cell types that we can use for administration. And really, we're still trying to understand where this might be the best or most appropriate application, but there's also a whole lot of research that's rapidly expanding around cell therapy. So those are kind of the three overarching ways that we think of within this field of delivering genetic material as the medicine itself: gene transfer therapy, gene editing, or cell therapy. And, I think it's important to note here that really this is a field that I know feels really recent because some of the advances have come more recently, but the science has been ongoing for over 50 years. And, in the last few decades, as I said, those improvements have been pretty rapid, but there is an enormous amount of research that has gone into getting us to this point, where to date, we now have a little over 5,400 gene therapy clinical trials across these modalities that have been planned, are ongoing, or have been completed for different diseases across these multiple approaches. So apologies for my dog. Good that we're recording it for posterity. But, the point is gene therapy is complex. And I think we are continuing to learn, but there's a lot of science that has gotten us to this point. All right. What are we hoping to do with AAV gene therapy in spaces like Pompe disease? Well, let's talk about, what current therapies typically are doing in terms of treatment intervention and protein level. So currently, for most folks, if you're on treatment, whatever that might be, you're going to receive treatment. Your protein levels are going to be up at a nice high level, but very rapidly, because of half-life and the realities of having a metabolism and pharmacokinetics, right? You're going to lose that protein fairly quickly to just your body sort of metabolizing it up. And so you have these peaks and troughs, as you can see in this first illustrated example. I think what we're hoping for in AAV gene therapy is that we will achieve a more consistent and durable level of protein expression, so that you will not be kind of at the mercy of these peaks and troughs, but instead we'll have a nice steady expression. And of course, our hope is that that may support better absorption, that not having fluctuating levels may just lead to more consistency in functional support of what enzyme is there in the body to do. And again, this is still being investigated, but this is the goal. So I'm going to take us back to our metaphor quickly here. Right, as we've already kind of covered, we do have this analogy of a functional gene as an instruction manual. And I want us to start to think of the vector as really the self-addressed stamped envelope of that instruction manual. So when we think about what we're trying to do with AAV gene therapy, we're trying to take an accurate instruction manual and put it inside of a vector, which is like a self-addressed stamped envelope, and I'll explain that a bit more. And we're going to put that into the mail, which is when we infuse it into your body, with the intention of delivering it to a specific address in your body. And in the case of AAV gene therapy, particularly for lysosomal disorders right now, that is the liver. So in a more true to scientific exploration of what that means, right, we're going to let's talk about vectors now for a minute. We're going to take that AAV virus, adeno-associated virus, and we're going to remove all of its viral information. And we're going to get rid of that so that now we just have that viral shell, which we tend to call a capsid. And into that, we're going to put that new piece of gene material in Pompe disease, right? That would be a new copy of GAA. We're going to put that into that little delivery vehicle. And now that new piece of genetic material inside that viral capsid is what we call a vector. So once we have built the vectors, we're going to infuse those through an IV administration into the body. And vectors naturally have affinity, what we call tropism, which just means preference. Viral vectors naturally have a preference for where they want to travel in the body. And part of why we select AAV is that it really likes to head to your liver. And once it gets to your liver, it's going to do what viruses do best, which is transfer its own genetic material into your cells. Now, if it were still a normal virus, that's when you would start not feeling so hot, right? You'd start getting the runny nose or the sort of achy eyes and think, uh-oh, I might be getting a cold. But in this case, right, that's not what happens because we've taken that viral material out. We've replaced it with the genetic material we want, and it's going to head to your cells and transfer that into the cells of your liver, which are also known as hepatocytes. And the goal is that then your liver, which is an organ that already likes to express proteins that circulate in the bloodstream, is going to start expressing that new enzyme. Your body will start making that, and it's going to push it out into your bloodstream, where it will then be in circulation for your body to use. So again, I just want to review, because I think we can never hear it probably too many times. When we're making vectors, we're using viruses because they're just incredibly efficient little delivery vehicles, right? Viruses have evolved over millions of years to be very, very effective at moving through our body, naturally gravitating towards certain types of tissues or organs. And so really what we're doing in AAV is we're taking advantage of that, but we're taking out that viral gene and we're replacing it with the functional copy of a healthy human gene that we want. So again, in the case of Pompe disease, that would be GAA. So a little bit more about why AAV. You know, AAV has a couple of features in addition to just being viruses with some of the attributes that I previously shared that we really liked, and that many of us in this research community really liked, which is why they're widely used. They're first of all not known to cause human disease. They also are a big enough envelope or storage vehicle, right? They can fit functional copies of genes fairly comfortably within that capsid. There's lots of different types of AAV. So you'll often hear about this as you'll see AAVs usually associated with a number. And those serotypes, right? And serotype just meaning preference in the body, where it wants to go more naturally in the body, are also a really nice feature, right? So AAV2 or AAV5 or AAV8 or 9, right? They all have slightly different serotypes. And, you know, sponsors are looking at those AAVs and specifically where they like to go as part of how they're thinking about delivering these medicines in the body. And so, you know, those are some of the reasons why AAV has become fairly common in gene therapy research for gene transfer therapy. I already shared, but the liver is just a really good candidate, particularly when we're looking at, you know, at something that we want to be systemically delivered, because it's already an organ that is creating proteins and distributing them out into the bloodstream. AAVs also already have strong affinity for liver cells. And your liver is also an organ that's just a nice sort of stable organ in your body. It doesn't have a lot of cell turnover as rapidly as other organs. So it's a good match when we're really wanting something to work over a long period of time. All right, so once we get that functional gene to your nucleus, where does it go? And I think we're going to talk about sort of two different approaches here for how researchers and investigators are thinking about gene therapy, and that is AAV, adeno-associated virus, which is what Spark uses, and there is also lentivirus. So I'm going to talk a little bit about both. And I want to say here, I think important to note about AAV and lentivirus also, a call back to gene transfer therapy versus gene editing versus cell therapy. I want to express Spark does not have a position that any of these are potentially better than any of the others. We think it's really important to educate on all of them because they all have really great applications. They all have important considerations when you are thinking about participating in research. And so we just think it's important that you understand all of them, which is why I'm speaking to all of them today. But I will get more specifically into what we work on at Spark in just a few slides. So AAV is what we would generally refer to as non-integrating. And what non-integrating means is that genetic material, as it's transferred into your cell nucleus, is going to turn into a sort of episome is what it's called. It's going to sit within that nucleus, but it's not going to integrate into your own DNA. Now, that's not true all of the time. Sometimes it does, but at very, very low levels. And most of the time, it remains what we would call non-integrating. So it sits sort of alongside your existing DNA. But what that does mean is that it is not passed on to new cells as cells divide, right? So as cells are turning over and dividing, that genetic material is not turning over with that new cell. So that means that AAV is really best suited for cell types in the body that divide really slowly and very infrequently so that we're maintaining that transgene expression. And important to note also, it does not affect any of your germline cells or your cells that are affected or passed down during reproduction. So the other approach that some of you may have heard of is lentiviral. And in lentiviral gene therapy, the gene is delivered into the cell's nucleus and it is integrating, and that is the intent. The desire with using lentiviral therapies is that would integrate into your existing DNA so that when your cells divide, that genetic material is passed down. So this is a really good platform or technology for when you are targeting areas where cells divide frequently, and you really want to preserve expression in those areas. It also is not passed down during reproduction. So again, we do AAV at Spark. I just want to call that out, but again, to say each of these have their really, I think, important roles to play in gene therapy research broadly and have important considerations when you're thinking about what might be right for you. So now we're going to transition to really talking about AAV gene therapy clinical trials and clinical research. So the first and I think most important part of participating in any research, but especially AAV gene therapy research, is an informed consent process that really ensures that you as a participant really understand what it is that you are agreeing to and what the potential risks to your participation for your own safety are. So I think all investigators, Spark included, take very seriously the importance of an informed consent process and really see it as one of the most important elements for how we think about participation at the community level. So I think it's really important that, the informed consent documents make clear in language you can understand what the protocol design is, what the objectives of the study are, how the study's designed, and any other attributes of the clinical trial, how long it's going to last, how many appointments you're going to go to. All of these details need to be made very clear in plain language. And an informed consent form is technically a document, but I think it's really important to emphasize that it is a process and it's a conversation that needs to be as long as you as the participant need it to be. So this is a document that you should review in detail, and you should ask questions with that person who is providing you the document. But you can and should also take it to your regular care provider or specialists within your care team that you really trust. You should talk about it with your friends and family and close members of your community that you trust. And really take your time with it, as it's really, really, really important that you understand fully what the clinical research is hoping to learn, and what the risks and potential benefits might be to you in participating. I can't emphasize enough, taking part in clinical research is your choice, and it certainly will not impact your current care. And that also it's important that you know that you have the right to withdraw from a trial at any time for any reason. So voluntarily agreeing to participate in a trial doesn't lock you into then being stuck. If at some point you voluntarily need to withdraw, that's also allowed at any time. So let's talk a little bit about what you can kind of expect to see in kind of the screening and inclusion criteria that we think about in AAV gene therapy clinical research. The first is typically we're going to have some restrictions around age. A lot of that is dependent upon the disease that we are studying. When does it onset? How does it progress? A lot of that will also be impacted by the stage of disease that we're being asked to really look at or focus on, either through our regulatory agencies or through unmet need. And, it will also just, potentially be tied to somewhat severity around, what we're trying to understand and impact. We're also going to look at your overall health history and specifically other medical conditions that you may have, as those could also impact enrollment criteria. So, certainly we understand in the rare disease space and in AAV gene therapy that, having other existing health conditions doesn't automatically exclude you, but certain things, particularly things that involve the health of your liver, since that's an area that we're going to be targeting for the therapy itself, as well as, other really complex health issues, are going to need to be understood and may impact your ability to participate. And then finally, neutralizing pre-existing antibodies. So you'll hear about these often referred to as NAbs, neutralizing antibodies, NAbs for short. And NAbs are antibodies that your body has already created due to prior exposure to AAV. So AAV is a really common virus in our environment. As I said, it doesn't make humans sick. So most of us don't know that we've been exposed to AAV because we had no real reason to ever feel that we had been exposed, but a lot of us have been. And each sponsor of clinical research is going to have guidance around a level of neutralizing antibodies that you can have in order to either be able to participate or that will exclude you from participating. And that's really because neutralizing antibodies can pretty significantly impact the efficacy of an AAV gene therapy if your neutralizing antibodies are really high. So a lot of people, tend to assume, which is very fair, that gene therapy research is going to be a couple of screening visits and then you're getting dosed. And that is not typical to what we see within the space. There's more typically a sort of observational study or a lot of pre-screening visits. You might have heard that also framed as that kind of lead up to actual dosing day. And a lot of that is to help us establish what we call a baseline of information on your specific body and your specific Pompe disease, right, in this instance, so that we have a really good, strong baseline understanding of information to then compare with what we begin to see after you have been dosed with a gene therapy study. So there's typically several weeks, could be as many as 6 to 8 weeks, right, of time where we're gathering a lot of observational data to then be ready and feel like we have a good picture of your baseline then to move forward at dosing day. So let's talk about what to expect as we get to actual infusion day. So let's first talk about dose, because it can look pretty confusing, right? The VG, kg, vig per kg, and I'm going to explain a little bit about how that dose is measured. So we dose in AAV gene therapy using this measurement. It's the number of vectors or vector genomes, which is what VG stands for, per kilogram, kg of body weight. And a dose, dependent on your kilograms, can contain millions or even billions of vectors. And that, I know, can sound like a lot, but I think we need to keep in mind that there are 240 billion cells in your liver on average. So when you have so many liver cells, we really need potentially billions of vector genomes in order to reach as many of those cells as possible on dosing day. So that's why, even though I know that number might sound big, it is proportional to how many cells you have in your liver, right? But we're going to determine your vig per kg dosing on your weight. And, depending on dosing, and each trial is going to have slightly different dosing, which I think is also important to mention. So that's something that you can be looking at as you're looking at different types of research going on in a space, right, are going to be delivered using different types of vectors. As I said, you see AAV5, AAV6, AAV8. You might also hear some things referred to as bioengineered, which might mean that they are combinations of AAVs, or in other ways being, engineered within a lab to typically attempt to kind of keep your immune system a little bit simmered down, so that we don't raise too many flags as we're infusing on infusion day into the body. But I think, in addition to what is going to be involved in dosing, I think other things that happen logistically on dosing day are one last health check, where we're going to check in one more time on a lot of just your basic vitals, all sorts of things. If you're a woman, a pregnancy test, are you running a fever? I mean, just some really basic health check assessments. You can anticipate that infusion typically takes about an hour. Again, that's somewhat dependent on your dose. And then you are going to be asked to stay at the study site, and how long is also dependent on the sponsor. You may be asked to hang out for a few hours and then come back the next day to do additional assessments. You may be asked to stay overnight for observation. It really just depends on the sponsor and how they've designed the study. So some of the things we're looking at, certainly, in terms of follow-ups immediately following dosing day, are going to be a lot of frequent appointments in those first few months really after dosing day, where you'll be coming in for a lot of assessment measures. So a lot of similar assessments to what you experienced sort of in that pre-observational or baseline period, we're now going to be looking at in a lot of the immediate follow-up to start to gather the data to compare to your baseline. We also have endpoints that obviously in research we are attempting to validate. And so we're going to be looking at assessments specifically around some of those endpoints. And endpoints are always listed in the protocol and should be very clearly laid out for you in an informed consent document, right? So measuring things like protein levels, looking at potentially biomarkers. I'm sure several of you have heard of this idea of biomarkers, right? So depending on what the endpoints are, you'll have certain assessments around those. And, you know, I think it's important to say you're going to have a lot of these. In gene therapy research, there are a lot of follow-up assessments, and it's a long-term monitoring environment. So we are mandated to make sure that we are following you, not just for 1 year or 2 years, but at a minimum of 5 years. And we try to be very thoughtful that that's quite a commitment. And so certainly you can anticipate that those visits will become less frequent over time. Those assessments will become less frequent over time. But I think it's safe to assume, regardless of the sponsor, if you're in a gene therapy study, the first year is going to be a significant commitment. And then from there, you might come in a few times a year or twice a year, and then maybe down to once or twice a year, right? It will begin to really taper. But that first year, you're going to be at a lot of follow-up appointments. And a lot of that is because we have a responsibility to make sure that as an entire industry, we understand the long-term safety and efficacy of gene therapy, right? So part of why we are mandated to follow participants in clinical research longer term in AAV gene therapy is that it is a newer technology, and we're really, as an entire field, trying to understand longer term impacts around safety and efficacy. So a few more things, and then we're going to get to questions, but things that I think are really important to keep in mind when you're considering whether or not to participate in clinical research, particularly AAV gene therapy clinical research, or any sort of gene therapy clinical research, really. I think make sure that your informed consent documents are a process and a conversation, not just a read and sign experience. These are complex trials where it's really important that you understand the risks, the potential benefits, and a lot of these other pieces as we move around the circle. The time commitment. You know, certainly these are, as I said, significant time commitments, particularly in that first year. But, you know, 5 years is a chunk of time. And I think understanding if the timing of that is going to work for your life is a really important thing to consider. Looking at that informed consent document, which will contain a schedule of assessments and essentially a map of what will be expected of you during that time, and really thinking about where you're at in your own life, thinking about where the trial is located. You know, most industry sponsors are going to support travel to the site location, but that still is time. You know, we will of course reimburse you for the costs, but it's still flights and hotel stays and sort of time away from your life. So those considerations need to be weighed, and, you know, how accessible is the trial location. You know, increasingly we are seeing more sponsors, I think, aware of the burden, bringing more home health visits into trial design, so that a lot of your assessments well, I shouldn't say a lot, so that hopefully some of your assessments can be done at home, particularly post-infusion day, right? And just understanding all of the testing and generally assessments that you're going to undergo, and making sure that you really understand and are comfortable with what's being asked of you. So again, I think important to emphasize that, really at this stage of gene therapy research across these platforms and across the industry, we're really still in a fairly early level of understanding. And what we're really, really trying to understand, certainly first and foremost, are these primary goals. Are these therapies safe? And are they effective? And is it an appropriate balance of benefit to risk? And at what dose is it an appropriate balance of benefit to risk? That's a lot of what we're looking at, particularly when we're in early stage gene therapy research trials. We're also, of course, looking at durability. How long will a single administration of an AAV gene therapy last? And I think here it's really important to call out that at this point, it is only one single administration, because we do not know yet how to successfully redose with AAV gene therapy. And that's due to those pre-existing antibodies. So even if you have low NAbs and are eligible to participate in a gene therapy study, it's important to note that once you have participated in gene therapy research, you have been dosed, and that means your body has been exposed to AAV gene therapy at levels where you will absolutely have antibodies that will make redosing at this point not possible. And this is something that the whole field is attempting to navigate and hopefully develop strategies and solutions for. But as of today, it is a single administration at AAV gene therapy, because we will not be able to redose you again. So we really want to understand how long these therapies will last. So these are things that you also need to really weigh for yourself. As you're thinking about participating in research, understanding the sort of balance of safety against potential efficacy, because certainly in phase 1-2 trials, efficacy is not even a primary endpoint. It is safety that we're really fundamentally looking at in those early stage trials. But absolutely, we also, of course, are seeking clinical efficacy. And I think, depending on what stage of research you are in, as you get into later stage trials, we are being held to demonstrate clinical efficacy, right? So you have to think about weighing the clinical efficacy benefits against some of the known safety concerns. And then finally, again, that durability of response. How long do we think this will last? And these are all really important questions and conversations to bring to certainly our Q&A today, although I can only answer certain aspects, but most predominantly your healthcare providers, your healthcare team, and so many of the incredible experts in this space that are doing research and providing clinical care. There's a lot that we are hopeful for with AAV gene therapy, but I think it's important to say that there are challenges. As I just spoke to, pre-existing neutralizing antibodies and development of antibodies after gene therapy administration mean that eligibility for gene therapy and to participate in research is going to be impacted, may be impacted, and certainly it does restrict our ability to dose the same gene therapy again. I think it's important here to pause and just say that if you screen out of one trial based on your pre-existing neutralizing antibodies, that does not mean that you won't be eligible for other trials. I think it's really important to look at your options and make sure that you're not assuming that you won't qualify when in fact you might, because as I said, sponsors have different windows of qualification for neutralizing antibodies. So don't let one screen failure in one trial, I guess, preclude you from considering other trials and talking about other trials with your healthcare team. And then a big part of what we are investigating in clinical research are possible risks, right? And I'm going to just list those here. So the current risks that we are exploring through clinical research are an immune response to gene therapy, inflammation at the sort of cellular level, as well as allergic or infusion reactions. And there are medications, and these are often designed as part of the protocol to really help sort of dampen or suppress immune response to encourage and, you know, sort of give gene therapy a chance to get in there and do its work. But this is a significant area that research is really actively looking at. One of the other risks is overproduction or overexpression of protein. You know, I think this is why you see somewhat conservative dosing with some companies, right? Because we just don't really understand long-term what the risks might be around overproduction or expression of protein that wasn't naturally meant to circulate in the body. And then, you know, certainly cancer. We have not seen cancers to date in AAV gene therapy trials at Spark, you know, but it is something that as a field we are watching for. That's one of those longer term safety implications that are really at the forefront of why we follow folks long term. And then there's a lot of unknown risk for pregnancy, which is why you'll see in a lot of trials that we require use of barrier protection for both males and females. Now, increasingly, this isn't just for males only anymore. So I just also want to, you know, make sure that some of the risks that are pretty common across all gene therapy research are discussed. Certainly, this is not a comprehensive or exhaustive list, and you know, this is why clinical research is so important. And this is what we're really all working on trying to understand. So these are the kinds of things that you want to really make sure you're talking about with your healthcare providers, and asking a lot of questions about if you are in the position to be considering research in that informed consent process. Again, we have a lot of hope in this field that over time, as we learn more and are able to apply that knowledge, that you know, we're going to see gene therapy treating potentially a whole host of diseases, but we're not there yet, right? We have a lot of work to do, and certainly it is partnership in communities and people who are willing to raise their hands and participate in research that drive a lot of that forward. So, you know, with that, I'll sort of close with, let's ask questions. Thank you so much for your attention. That's you know, an email address that you're always welcome to reach out to me through with questions. So if you don't get a chance to ask one today, don't be shy, send them my way. But I'm going to stop screen sharing and come back on video, and just make sure that we address questions.
On behalf of the AMDA, I want to thank you again, Jacose, for a really great, informative presentation. And I think, you know, it really provided a lot of good information on what we should be thinking about in terms of informed consent and expectations in a trial, just all around, not just for gene therapy. So thank you again. And with that, this concludes today's webinar. Thank you for participating.
Thanks, everybody. Thanks for having me.